welcome to your very own BYJU'S 6th, 7th and 8th grade channel. I'm your teacher Aishwarya, and in this video we are going to be solving top 10 most important questions from Microorganisms Friend and Foe, where we will specifically look at all the important questions from beneficial microbes. So I hope all of you here are excited for today's class, and very quickly I hope that my audio, my video and my screen and whatever I'm writing on the screen is visible to all of you. If it is, very quickly give me a thumbs up in the live chat to let me know that we are good to go. So I can see that many of you are here already, so I welcome all of my students to the class. Now I can see many of you are here. Good evening to Jitender, Manisha, Darkstar, Rajendra, Savani, Purvi, Anushka, Sharvi. Welcome to the class. Welcome, right? So now of course I can see many of you are here. Yes, already questions are coming my way. I will be discussing it. So how we're going to do is, first we will do a chapter summary. So whatever I have covered so far, I will summarize it, followed by which we will be looking at some 10 important questions, right? So they are going to be objective questions, and of course we may not have polls per se, but nonetheless you will be able to answer. So good evening all of you. Good evening. So very quickly, I hope all of you are excited for the class. Yes, I can see that Anushka, Preeti, Ashish, wow, so many regular students, so many new students. I welcome all of you to today's class. Yes? Okay. So now, of course, very quickly, yes, I will answer. Ansh, of course, I will. Ansh is very punctual. Ansh will make me also punctual in case if I am slacking off a little, right? So many of you are saying you've got good marks as well in science. Good evening. Very excited as well. Awesome, right? So now, of course, we're going to get started, but very quickly, students, you know that when you come to my class, there are a few things to keep in mind. So my regular students will tell you what you need to do, but for everyone else who are new, right? First and foremost, in my class, please make sure you have your notebooks and you have your textbooks ready with you, along with which you will have a pencil, pen, whatever you want it to be, right? Pencil, pen, sharpener, everything, you keep it ready. Don't go in between and take it. Yes? Secondly, also have your water bottle as well, right? Please have your water bottles ready, because I don't want you missing some class in between because you forgot to have water. Now, most importantly, as we all know, what are we doing today? We are doing a chapter summary. And then, of course, we are also going to be having important MCQs, multiple choice questions. So when I do chapter summaries, there is going to be a time when I will be teaching. Now, when I am teaching, kindly avoid saying the following things. Ma'am, you are ignoring, right? Ma'am, I'm writing this down because this is your popular dialogue. You are ignoring me, right? Ma'am, you are not taking my name. Ma'am, you are not answering my doubts. Understand that the chat is still yet to be set up on my screen, so I'm looking at my phone, right? And secondly, of course, very importantly, what I need you to keep in mind is that I will take your doubts when I do a doubt board section, right? So everyone, I'm going to be taking a doubt board section. So there will be parts where I'll only clear your doubts, yes? Now, understand that sometimes all of you will have similar doubts, okay? And if you pay attention to my explanation, half your doubts are also going to be cleared then and there, yes? So if you follow these things, today's class is going to be seamless. Otherwise, many of you are here in the live class. So many of you are here today after a very long time. Feels very nice to be teaching 100 plus students in the live class, right? Hoping that one day it's going to be 300 and 500 live class students that I would want to teach. But good, yes? Now, of course, let me tell you one thing. In case if you want a short, crisp video, I've also done a recorded bit as well in Concept Bites, which you can watch, and trust me, it will be very simple and easy for you there. Yes? Okay. Are we all good to go? Are we ready? Yes? Just give me one moment as I set it up. Okay. Okay, students. Very good. Amazing. Amazing. Right? Yes. No, nobody says sorry. Everybody's like, ma'am, please speak slowly. Yes, I will keep that in mind. So quickly, students, if you are excited, hit the like button on this video. Do not forget to like the video. Do not forget to hit the subscribe button. You are all saying that you are all excited for the class. That means you've been waiting for it for a long time. And it also means you should like the video. Yes? Okay. Yes, I will tell about Leeuwenhoek. Very good explanation. Very good point. I will tell you about Leeuwenhoek, right? Okay, now we're going to get started with the chapter summary, right? So now, of course, when we talk about microorganisms, friend and foe. Foe means enemy, right? Friend means we know what friend is. But of course, when we talk about microorganisms, this particular chapter has three broad parts. We talk about what are microbes and the classification of microbes. We see that how microorganisms are beneficial and how some microorganisms have harmful effects. Now, of course, today we are going to be covering these two aspects, which is classification of microbes, right? And what microorganisms are basically. Now, when we talk about microorganisms, right? We are living organisms. You and I, you, me, plants, animals, everything is living, right? And what do we say when it is living? It has the ability to grow. It has the ability to maybe produce energy, eat food, right? This is what we consider as living organisms, right? But we also see that there are some living organisms which are not visible to the naked eye. That means if I just look at the palm of my hand, right, what will I see? I will just see my palm. I won't see anything else. Yes? But if I bring in a microscope, if I bring in an instrument and if I zoom closely into it, I will see that there will be many tiny organisms, right? So these tiny organisms is what we call as microorganisms. Yes? So micro means we require a microscope. Yes? स्टार आर्टिस्ट बच्चा आज क्लास पे ध्यान दो, फ्रेंड्स बाद में बनाओ, राइट? So here, as you can see, it is microscope. Yes? Now the microscope that was there, right? The microscope that was there, of course, has undergone a lot of iterations. But the first primitive microscope which Ansh has brought about was discovered by Anton van Leeuwenhoek, right? Now Anton van Leeuwenhoek has a very interesting story. You know, Anton van Leeuwenhoek was actually not a scientist. Anton van Leeuwenhoek was a textile vendor, okay? So what he was doing, he was selling, he was a businessman, he was selling textiles. Basically, he was selling clothes. But one day he was very excited to know how his clothes would look like. Now, if I zoom into my clothes and I see what will happen. So he started to, meddle around a little bit and he started using some, magnifying glass to look closer and closer. So then he saw that, his clothes had fibers and everything. Then, of course, later on, right? Later on, what happened? He started to observe other things. He wanted to see how the pond water will look like, what a leaf would look like. So he started to go and he started observing everything under the microscope, right? So that where he will be able to help. That's where he was able to build the first primitive microscope, right? Yes. Virus ka, I will tell you, Pinky. Again, I always tell you in the beginning of the class, this is also what I said. Ranjit, can we also have that on? So this is what I meant, right? Pay attention. This is how exactly he was able to do this, right? Yes, Talat, Bacha, I think that you probably, just pay attention, trust me, you will be able to understand. Ma'am, how to remember his name? Remember Anton, enough, right? Microbes he discovered first were actually bacteria, right? Okay, if screen is not clear, go to settings and make it better, right? You can improve the settings. No mentee today, no mentee, poll questions only, right? This is some extra information for you, that's all. Now, let me tell you, somebody has doubt. Ma'am, microorganism, microbe, everything is there. Is microbe and microorganism the same or is it different? Microbe and microorganisms are the same only, right? They are not very different. Interchangeably we use. Yes? So in this particular case, we see that there are various types of microorganisms, right? So we see that microorganisms just like animals, right? It's not like we have only one kind of animal. We have so many varieties of animals, right? So we see that there are different types of microbes as well. So broadly, you have bacteria, fungi, algae, protozoa and virus. Yes? Many of you here want to know what is virus, if virus is a microorganism or no. For those of you, wait 5 minutes, I will be explaining, right? Now, bacteria that are there are basically unicellular organisms, right? Which means that their body is made up of one single cell. And we see that they come in different shapes and size, right? Now, there are some bacteria which are rod shaped. So, for star artist who's asking me, ma'am, what is bacilli or what is bacillus? Rod shaped bacteria have the name bacillus in them. If they are circular or spherical, spherical in shape, we say that they are cocci, or we say they are coccus in shape, right? Vibrio means they are comma shaped, and if they are spiral shaped, we say that they are spirilla. This is some extra information. Ansh, if you keep spamming, it becomes difficult. At one point, I will only see your doubts. What about the others who have doubts? No? Very important to answer everyone's question. Ma'am, what's there in the microscope that we are able to zoom in? Lenses are there, right? So you've learned about this in light chapter, that of course with the help of lenses, and you'll be able to zoom in and zoom out, right? So you see that the way the lenses are arranged and various other factors help us use this. Yes? Okay. So now, of course, this is all about the different types of bacteria. Similarly, we have fungi, right? Now, what are fungi? Fungi could be made up of one single cell. They could be unicellular, or they could be multicellular, right? So, unicellular organisms include yeast, yes? And while on the other hand, multicellular organism is mushroom, right? And in these cases, we see that normally fungi are found growing in dead and decaying matter. Now, does not mean that they are always found growing on dead and decaying matter. It is just that these organisms are naturally, yes, they are naturally found growing in dead and decaying matter. So far, are we all clear? Phosphate is basically a chemical compound which has phosphorus and oxygen in it, Mushkat. Okay? Talat, I know you have not understood, which is why I am teaching from the beginning, right? Yes. Ma'am, can yeast be multicellular? No, it cannot be multicellular. What is spiral? Spiral shaped, right? That is spirula. Cocci means spherical, spherical ball-like structure. Yes? Ma'am, is fungi concluding? Ma'am, virus is microorganism. I'm getting to that. Virus ka you wait. But so far what I have taught you, are you clear? So far, I know there are many things that you don't know. But so far, are you clear? Ma'am, you are telling fungus is microorganism, but I am able to see. Good question. Love that question. See, fungi that are there are actually not all fungi are categorized as microorganism. Good catch. Now, of course, I told you that multicellular organism may you have fungi. There are fungi which are very big and very visible to the eye, right? But you have bread mold, right? You have bread mold. And you see that bread mold that is there is multicellular. Yes? But when you look at it in the bread, no? We've learned about bread mold in our 7th standard. When you look at bread mold, all you will see is one chotu patch, right? You will only see one patch, and that is all you are able to see. So this right here, right, this bread mold, that is an example of a multicellular organism which is not visible to the naked eye. But yes, mushroom is an edge case which is not a microorganism. You could say it's a macroorganism, right? Okay. So now, of course, okay, I will finish syllabus. Don't worry, right? Yes, ma'am, in my book there is written 5 types. I am getting to it. I just came to the 2nd type, right? Okay. Awesome. Bacteria discovery, I just told you it was done by Leeuwenhoek, right? Lactobacillus is an example not of fungi but of bacteria, right? Yes, some fungi are, it means that very good Jitendra, some fungi are visible while some are not, right? Then of course we have protozoa. Now protozoa are unicellular organisms, right? So again they are unicellular organisms, but they differ from bacteria because these organisms are little bit more advanced, right? So here we see that there is amoeba, paramecium, somebody was asking about euglena, right? So euglena that is there is like a spindle shaped, which means it is broad in the center, it is narrow towards the end, and they have a long, not a tail, but they have a long whip-like structure. So ye whip, what you could say, this whip that is there could be like how if you had a belt and you are moving this belt back and forth, right? So this right here is how it will be able to move. Yes? So this is here all about, this is about euglena, which is an example of protozoa, right? Similarly, paramecium is a slipper-shaped organism, and amoeba that is there does not have any definite shape, right? Then of course we have algae. Now algae is a very wide category, okay? Because again, like mushroom, like your fungi, which could have unicellular and multicellular, algae also are unicellular, some of them, like your chlamydomonas. While Some are very big in size, like giant kelp, which is visible to the naked eye. So there is only one category of organisms that come in as microscopic algae. Then, question of the hour. Ma'am, virus is fungi or no? How many of, virus is microorganism or no? How many of you have this doubt? Ma'am, I don't know about virus. This virus is very confusing, right? Ma'am, algae is both unicellular and multicellular, right? It is both. How many of you have this question of the hour, right? No, ma'am. Bye, ma'am. Okay, bye. Yes. This time Ante is in October, right? Very good, very good. I have not even reached to location. I have just come to the first part where I have categorized this, right? Coccus, cocci is plural, coccus is singular. Ma'am, it is boundary line. Okay, that means students who are giving me the answer have watched my video. Students who have not giving, who are still confused, probably you have not watched it yet, right? Yes. So now, of course, when you talk about viruses, see, viruses are microscopic. What do I mean by microscopic? That means that I cannot see a virus. If one virus is floating around here, I will not be able to see it. I require a microscope to observe. It is microscopic. But you will not say it is a microorganism. Why? Because virus is somewhere between living and non-living, right? So this right here exists somewhere between living and non-living. Now what do I mean by this? Now for viruses, right, I told you that there are some criteria for an organism to be living. So in the case of a virus, so let me just draw some random virus. Imagine this is a virus. For this virus to be living, it needs to reproduce, it needs to make more of itself, it needs, energy, right, in order to survive, right? Now in all of these cases, we observe that when it's floating around, it is fine. It is not doing any of this. It is behaving like a non-microscopic organism, right? Or I will say it's behaving as a non-living organism. But when it encounters a host cell, right? Like for example, it could be maybe a cell in our body, a cell in a other animal's body, or it could be a cell in a plant, right? When it encounters a host cell, what it will do? It will hijack, right? So just like how if a robber comes into our house, he will take everything and go, right? All the money he will take and go. Just like that, the virus will come into our cell. It will use everything that is there in our cell, right? And we see that because of this host cell, it will utilize all of it in order to reproduce, to get energy and for all of that. Which is why I will not call virus as a microscopic organism. I will not call it because it is somewhere between living and non-living. But yes, virus is microscopic. Are you all clear now? Are you all clear? Yes? Does virus have a definite shape? If then what shape? It has different shapes, right? It has like a eye. So basically, if you see, some of them are spherical, some of them, if you see, have like a octahedron kind of thing, right? So they have like it has a specific name, but I'm not going to get into that. But it will be like this. Something of this sort. Yes? Okay. Living cell, non-living outside. Exactly. Okay. Very good, all of you. Very good. Ma'am, your opinion on this chapter's weightage. High weightage chapter. Very, very important, right? No problem, Purvi. Come back and watch the video, right? Chlorella is an example of algae. Yes, it is. It is. I am getting to it. I am still in classification of microorganisms. I told you clearly that I will get to all of this, right? Thank you, Saksham Bacha. Thank you so much. Very good, Kavya. Very good. Yes. Ma'am, how do microorganisms make more of themselves? See, we will learn in reproduction chapter. So there are various ways in which a single organism will make more of itself, and we call this as asexual reproduction, which we will do. Why virus is not bacteria? Because there are various categorizations for bacteria, right? Bacteria is a living organism. Virus will not come under bacteria. It is separate. Yes, Shanmukh, I hope you are clear. Where do virus reproduce? Inside a cell of the host, right? Yes. Ma'am, Volvox is unicellular or multicellular? Oh, very good question. Volvox that is there is made up of many tiny compartments that are there. So, kind of considered as multicellular, right? Yes, but it's a microscopic organism. Ma'am, I saw your old videos and I will understand. Yeah, you will actually, right? And what's the recorded videos also. I will teach you asexual reproduction and everything in that chapter. Don't get distracted, okay? Chlamydomonas is an example. I know, it's okay. Moving on. I have explained it. Right now, I always tell you, I take doubts. If you're not paying attention, you will go idhar-udhar, right? Yes, we will do Olympiad. We will do. Ma'am, can virus act like a cell, like HIV? HIV is an example of a virus. HIV is human immunodeficiency virus, which is an example, right? Yes. Now we will move on to understanding more about microbes and their uses. But before that, let me tell you. We saw that there were so many different kind of microorganisms, right? So many different types of microorganisms were there. And we know that microorganisms are those which are found in different parts, right? Or we find them in different places. It's not that microorganisms are only found in land or water. We see that there are microorganisms found on land, especially in air, water, soil, and we also find them in some extreme conditions also, right? So in extreme conditions where there is a lot of maybe high amount of salt or maybe high temperatures, right? We know that these microorganisms have the ability to survive in such conditions as well. Yes? So with this, of course, right, as we all know, we know that microorganisms are omnipresent. That means that they are found everywhere. But in one particular case, what we need to understand is that microorganisms most often than not have a very bad image, right? Because in our lower grades, we have all learnt about germs. So when we talk about germs, we know that these are organisms which can cause harm to our body. And later on, we will understand that disease-causing microorganisms are called as pathogens. What are pathogens? Pathogens are disease-causing microorganisms, right? So these are the disease-causing microorganisms that are there. But many of them, right, are beneficial to us. So we will understand, yes, we will understand how they are beneficial. They help us with food production, right? So let me just go back to that. So in this particular case, we see that they help in food production, they help in the medical industry, they help in agriculture, so on and so forth, right? So let's understand in food production. Now in food production, of course, they help in manufacturing dairy products, in baking goods, wine and vinegar as well, right? So now we will understand this one by one, okay? Now very quickly, I think I will just go on to the latter slides. I'll go back again, sorry. So now let's understand this particular part, right? Now in dairy products. So now here in dairy products, we have learnt about curd formation, right? Now, how does curd formation take place? How many of you are clear with this topic, right? How many of you here are clear with this particular topic? Can you tell me? I am going to revise this part once again, right? So, when you talk about curd formation, we know that in the case of curd formation, we know that there is milk, right? So, we see that there is milk and we will add little bit of curd to it. We will keep it overnight, and what is going to happen? Overnight, we will see that there is formation of curd. Now, this happens because inside this curd, we have lactobacillus, right? So, here we see that we have lactobacillus. Now, what is lactobacillus? Lactobacillus is a rod-shaped bacteria. Students, if you want notes, you write it down as I am teaching you, right? It is a rod-shaped bacteria which is found in the curd, right? Now, of course, we see that once we add this curd, right, the milk in itself has something inside it, right? Milk has various proteins, but it also has lactose. Now, this lactobacillus will use this lactose in order to make more of itself. And when it utilizes this lactose, it will result in the formation of lactic acid. So here your lactobacillus is converting lactose to lactic acid. Then what will happen? This will eventually, because lactose has become lactic acid, all the proteins that are there will start undergoing some changes, because of which we see that your milk that is there turns into curd. Are we all clear? Ma'am, does lactobacillus contain lactic acid? No. Lactobacillus will convert lactose to lactic acid. Yes? Quickly to recall, Curd formation takes place with the help of curd, right? Inside the curd, lactobacillus is there. Lactobacillus is a small rod-shaped organism like this, right? It's a bacteria. What it will do, when you mix the curd and milk together, your lactobacillus has entered into the milk. Inside the milk, what is there? Lactose sugar is there. It will use this sugar and then it will convert it into lactic acid. When it will convert, protein changes will happen. When this takes place, milk will become into curd, right? So I hope all of you are clear. See, I am not ignoring anybody. ऐसे very simply in easy words I am explaining, right? Are we clear? Yes. Any doubt so far? I know, but see, with microorganisms you require more and more practice, right? Yes. See, I can give you questions, but I want you to make sure you understand. Very good, Saksham. Yes. Haan, bacha, what is the doubt? Ma'am, this process is called fermentation. Yes, this is called as fermentation and we specifically call it as lactic acid fermentation. Ma'am, define protozoa. Protozoans are, this is for Nirupa Barik. Nirupa Barik, protozoa is a microorganism which is unicellular and we see that they have only, right? They have only a cell membrane, right? And we see that they have the ability to move around. Is Euglena autotrophic? Atharv is asking me. Euglena is autotrophic, shows autotrophic and heterotrophic kind of nutrition, both. Ma'am, rod-shaped bacteria is bacillus or lactobacillus? Rod-shaped bacteria is bacillus. Example of bacillus is lactobacillus, right? What is inoculum? Inoculum is a fancy word for the curd that we are adding, right? Yes. Lactose, Jitender, is a sugar. Casein is a protein, okay? Yes, all right. Hepatitis and everything in a different day. Yes. About half an hour more, probably, right? Now, next up, we have production of confectionery and basically wine, right? Now, this, of course, uses another kind of microorganism, which is going to be yeast, yes? Now, what is yeast? We know that yeast that is there is a unicellular, this is a unicellular fungi, right? And we know that this is also, the scientific name is Saccharomyces. Now, basically what happens is that yeast will facilitate a process called as fermentation. Now, what is fermentation? Fermentation is a process wherein there is conversion of sugar into alcohol, right? Alcohol and carbon dioxide is given out as a byproduct. Now this right here, yes, this is a process that we use in order to make bread, in order to make pastry, right, or even bhature, right? All of this is the process that we will use, right? So let's take the example of bread. What exactly happens during bread making? Now when you make a bread, what will you do? You will take flour, you will take water, right? You will add some sugar to it and you will add yeast, right? Now in this particular case, what will happen when you mix it and you make it into a dough, right? You will make it into a dough. You will take it nicely, you will make it. And what you will do, you will cover this with a muslin cloth, right? Or any cloth, you will keep it for some time. Now inside what is going to happen, let us see. So inside, now your yeast is there and your sugar is there. Now your yeast that is there is going to increase or it will multiply, right? So it will multiply and make more of itself. Yes, it will make more of itself. Now when it makes more of itself, it will utilize all of this sugar, right? And it will convert it into alcohol. Now your carbon dioxide is also getting produced. Now what is carbon dioxide? Carbon dioxide is a gas, right? So the gas will try to escape, yes? So when the gas will try to escape, we see that it will try to go. Now when this happens, what will happen? The dough will increase in size, right? So we see that the dough that is there will increase in size, or we say that it will increase in volume, right? And what do we call this? We call this as risening of dough. Why does risening of dough happen? Because of carbon dioxide. So far, are we clear? Yes? So far, are we clear? I have seen bacteria under the microscope. I have seen lactobacillus only under the microscope. See, I am not asking about other things. So far, what we have discussed, are we clear? Viruses between living and non-living. Saccharomyces, very good, Shanmukh. Saccharomyces cerevisiae is the example of the yeast, right? Good. Ma'am, why curd is important in rava? I am not sure about that. Yeast is a fungi. Yes, yeast is a fungi. Okay? Good. So now rising of dough has happened. Now when you take this dough and you put it in the oven, right? You will bake the bread. So when you bake the bread, all this alcohol will evaporate. All the alcohol will evaporate, and whatever carbon dioxide is there, it will expand, and this is what will give rise to the texture of the bread, right? So this right here is what we understand as fermentation. Now this topic is very, very important because they will ask you questions from this. So are we clear? Are we clear about fermentation? I hope that it is clear because exam point of view mein this is super important, right? Crystal clear. Awesome. Antibodies, everything I will tell you now. Let me get to that part. Don't worry. Ma'am, please answer this appropriately. Anti-preparation will start very soon. We are going to start very soon. Ma'am, what is most important in leguminous? Root nodules mein rhizobium is there. Symbiotic bacteria, I will teach you. Why is curd healthy? Because we see that lactobacillus is not only present in curd, but it's also there in our digestive tract, in our gut, right? So there are some healthy microbes even in our gut which is necessary for us. Yes? Fermentation, everything I have explained. Okay. Now we will move on to the next one, which is antibiotics, right? So now we see that antibiotics that are there, it comes under the next category, which is in the field of medicine. How many of you have taken antibiotics? I will discuss nitrogen cycle also. Don't worry. I will discuss everything as long as you pay attention. Yes. Okay. Now, antibiotics, all of us have taken, including me, right? So now what are antibiotics? So let's break the word now. Anti means against, right? Bios or biotic means life. So here antibiotic means against life. So antibiotics that are there are basically chemical substances that are obtained. That means I get my antibiotics from microorganisms. Okay. So I'll get it from some other microorganism. It could be a fungi, it could be a bacteria, right? But I will use this chemical substance in order to kill or inhibit the growth. So I will either use it to kill my disease causing microbe or I will use it to stop. So I will say stop the growth of disease causing microbe. Are you clear with this? Are we clear? Very easy. Yes? So now of course this is what we understand as antibiotics, right? Now the first antibiotic was discovered by Alexander Fleming, and of course the antibiotic he discovered was penicillin from a fungi called as Penicillium notatum, right? So this was the first antibiotic, right? Then of course we see that other examples are streptomycin, erythromycin, tetracycline. And of course, we see that it is not only used just for human beings, but also to treat animals, protect them. It is mixed with the animal feed, right? Then of course, we see that there are various disadvantages There are various disadvantages also, or rather I would say there are some precautions to take in mind. What are the precautions? First and foremost, we should eat antibiotics only if prescribed by the doctor. Should not go to the medical store and say, give me this antibiotic, right? So why should we take it with the suggestion of the doctor? Because antibiotics have an impact on the growth of microorganism. But they are chemical substances. They don't know if a microorganism is good or bad, right? They don't know if it is good or bad. Now we know that there are various microorganisms which are present in our digestive tract also, right? There are various organisms which are present in our digestive system. So what will happen? If you eat too much of my antibiotic, then what is going to happen? It will kill the good microbes, right? All the good microorganisms in my body will die. But if I eat too less, the disease causing microbe that was there, maybe it could be some typhoid causing one, right? The disease causing one, now what will happen? It'll find a way. Oh, this person is eating this antibiotic, I can do well, or I can, escape this antibiotic. So too less of it will actually make the antibiotic ineffective, right? So if they say eat it for 5 days, complete it, right? Complete your course. Don't be like, ma'am, I ate antibiotic for 2 days. Third day I was feeling very good. I will not eat antibiotic. Don't do that. Even if you have finished it for 2 days and you're feeling better, but you need to complete the course. Yes? Ma'am, how is it? Ma'am, if it is obtained from microorganisms, very good question. Because there are some chemicals. So there are some chemicals, right? So here the question was, ma'am, if we get an antibiotic from a living organism, from one microbe, how am I using it to kill another microbe? Because it is chemicals which are released by, let's assume, microorganism X, right? Microorganism X is a fungi which is producing some chemical. Now this chemical which microorganism X produce is going to be harmful, right? It's going to be harmful for maybe bacteria Y. So it is kind of like how we say, no? Can we eat all plants? I have a question for you. Can we eat all plants? Yes or no? Can we eat all plants? Yes or no? What do you think? Okay, now tell me why. Tell me why you cannot eat all plants. No, everybody's on. Not all are edible. Exactly, right? Because some are harmful. But you will tell me, but I can tell you, no ma'am, plants are good. Plants are good for us. Plants are giving oxygen. All plants should be good. That's exactly the logic of microorganism. Although all microorganisms are there, we see that there are some chemicals which are produced by microorganisms, say X, which is not good or which is going to be toxic for another microorganism, right? So it is the same logic that we are using. So this was all about antibiotics. Next, we have another one which is vaccines, right? So now, honestly tell me, how many of you are confused about vaccines? Yes? How many of you are confused about vaccines? And what do antibiotics do? They kill or they stop the growth of disease-causing microorganisms. Ma'am, me. Many of you are confused, is it? Hooray! Awesome topic this is. Works against, penicillin is normally used for bacterial infections that are there, right? Hooray! Vaccine is my favorite topic. So let me tell you about vaccines, right? Now vaccine was discovered by Edward Jenner. And of course we see that vaccines that are there, right? What are vaccines? I'll go back to this slides. Vaccines are a chemical preparation, right? A chemical preparation of dead or weakened microorganism or microbes. Yes? So I have studied biotechnology, right? And of course in this field of biotechnology we see that there are applications, there are methodologies in which we can prepare these vaccines. Yes? So what we do, there are laboratories which are set up, and of course safe practices laboratories which are there, wherein we see that these microorganisms, their capacity to cause a disease, right? So here when I say dead and weakened microbe, it's not like I took some lactobacillus from the curd, right? Not like I took some lactobacillus from the curd and I made vaccine, right? So a vaccine is a preparation of dead or weakened microbes, right? And these are microorganisms that have the ability to cause a disease, right? So these are not some any random microorganism. These are microorganisms that can cause a disease. Yes? So these are what we call as dead and weakened microbes, right? So what is the difference? Vaccine is a preparation of dead and weakened microbes, right? See, pay attention to me, because if you don't pay attention, this can be little tricky. Okay. Now, antibiotic is a chemical substance which we get from a microorganism to kill it. But here, that is a chemical substance. Your vaccine is a preparation of dead and weakened microbes, right? And what do? We make this for a reason, right? We do this so that our body. So now if you see, earlier in the days, normally, how I'll do this with the help of an example. How many of you have played chess? How many of you have played chess or any sport to honestly, right? How many of you have played chess or any sport? Can you tell me? Everybody has played sports, right? Now, when you play chess, or let's say if you play basketball, because I've played basketball, I can give you that example. When you play basketball and you're going for a match, not just in PT period you're playing with your friends or everything, but we're talking about when we go for an actual match or a tournament with other team and you're in a competition, what happens? Do you just go like this without any practice? No, right? You will go with practice and everything. But still, if you are in team A, and you are competing with team B, you will see, if you knew how team B played, probably let's say team B had a match with team C, and you are seeing team B play. When you see them play, you will be like, okay, this player is very strong. Oh, this person can do this. So this will give you an added advantage, no? You'll be like, I've already seen how team B plays. So when I am going to play against them, I will know exactly what to do. Yes or no? Yes? We know what to do, no? If we have already seen team B play, right? Yes or no? Arey, nobody's listening to me. Live chat pe, they are all doing their own thing. Now imagine, right? You imagine, you didn't know how team B played. You have no clue how they played. And during the match, you are playing them. Chances of you maybe making some mistakes, or maybe losing the match is also very high, right? But if you have a strategy in mind, it makes things easy, right? So this is how we are going to have, or this is the ideology behind vaccines, right? So vaccines that are there are basically chemical preparations, right? So we see that these are chemical preparations which include your dead or weakened microbes, right? Now we do this wherein we will inject it. So injection is either injecting into my bloodstream, right? So injection is a way in which I will take the vaccine. I will inject it into my body, or in some cases I can orally take it also. So the difference between a vaccine and an injection is how it will enter. Vaccine is the preparation, right? So we see that this right here, once it enters into our body, right, our body's defense system will get activated, right? So our body's defense system will get activated. So which means all the defense WBCs and everything will get activated, and then they will produce some Y-shaped proteins, right? So we see that they will produce Y-shaped proteins called as antibodies. Yes? So what are antibodies? Proteins produced within my body. What are antibiotics? Chemicals which are produced by other organisms. Are you clear? Yes? Are you all clear? WBC is white blood cells. What is antibody made up of? Proteins, right? Yes. Okay, very good. Exactly. So this is how we do it, right? Y-shaped proteins. How was vaccine discovered? By Edward Jenner when he was trying to find a cure for smallpox, right? Yes. Antibodies and vaccines ka also we are clear, right? Is there any natural vaccine? Good question. Is there a natural vaccine? So normally if you have not got a vaccine and you have fallen sick, right? Maybe one disease causing microbe will enter into your body, you will fall sick, your body will fight, they will produce these antibodies. But at the end of it, naturally your body will develop a it will develop a resistance to that. So these antibodies that are there, we see that they are Y-shaped proteins, okay? And we see that these are highly specific, yes? We see that they are highly specific, which means that if there are thousands of disease-causing microbes outside, no, our body has the ability to produce thousands of antibodies, right? That is what we mean by they are highly specific. Then, of course, we see that in this particular case, we also see that our body has the ability to remember. So if thousands of microbes are there, our body has the ability to remember so that they know which exact antibody they have to produce, right? Ma'am, a vaccine is made by dead and weakened microbes. How can our body fight bad microbes? So basically, like I said, no, antibodies are highly specific and our body has the ability to remember. So when I put a dead and weakened microbe into my body, I am not really getting a disease, but it is also activating my defense system, right? It is also activating my defense system in my body that is telling that, hey, you know what? This particular microorganisms look deadly. So it will make this antibody. It's kind of like already preparing, right? It's already preparing. Very good, very good. Yes. So of course with this everyone we see that normally we get vaccines to get protection against diseases, right? Then of course last we have the impact of microorganisms in the environment. So when we talk about this, we know that it helps facilitating decomposition where dead and decaying matter is broken down into simple soluble forms, right? And of course there is contribution in the nitrogen cycle. Now nitrogen cycle I will explain because I have a question based on nitrogen cycle, right? But of course very quickly we will move on. I realize 40 minutes I have only taught and I forgot to take questions, right? So jaldi we will do the questions also. We will wind up in around 20 minutes, right? 20 minutes may I will wind up. Okay, are we ready? 3, 2, 1. Time to start. Yes? Are we ready? And I hope all of you have hit the like button on this video, right? Very important that you hit the like button on this video because I can see that many of you are here and very few of you have come to the class, right? Okay, moving on to question number 1, right? Question number 1 is, which among the following will reproduce only inside a host cell? Which among the following will reproduce only inside a host cell? Lactobacillus, penicillium, retrovirus, or is it yeast? We are talking about which of the following. It is not even telling which of the following organism. Focus on that, right? Which of the following organism will reproduce only inside a host cell? Yes? Ma'am, what Nirupa is asking me, ma'am, what will happen if I put lactose and yeast? See, lactose, let me tell you, yeast has the ability, there are certain specific chemicals which the yeast will produce that has the ability to break down other sugars, right? In the case of lactose, you never know. Maybe it does not have that. Probably it will remain, it will not act on it because it does not have the chemical substances to convert lactose into lactic acid, right? So polls are not available at the moment, right? So very quickly students, it's okay, poll is not available. I'm really sorry. It's okay, you answer like this. So in this particular case, we see the answer is virus. So somebody asked me what is the definition of virus? Virus is a microscopic structure that has the ability to reproduce and survive only inside the host cell. So correct answer here is option C. So 1 mark for all of you who have got the answer correctly, right? Moving on to the next question. What causes the dough to rise when yeast is added to it? A, increase in temperature. A, increase in the amount of substance. C, an increase in the amount of water released by the yeast cells, or the release of carbon dioxide. Hello Siddhi, hello. Very good all of you, very good. Yes, exactly, right? So in this particular case, it's very simple. When yeast is added to the dough, we know that the fermentation process is going to happen, right? And when fermentation happens, carbon dioxide is produced, and the release of carbon dioxide will cause increase in volume, hence causing the dough to rise. Yes? Moving on to question number 3. Curdling of milk is caused by what? Rhizobium, lactobacillus, paramecium, or yeast? Array! Super easy question. Yeast is not a virus. Yeast is a fungi. Very easy. Curdling of milk, which results in the formation of curd, is because of lactobacillus. Very good students. 3 out of 3 for all of you. Let's move on to the next one. Medicine that kills or stops the growth of harmful disease-causing microbe is called as what? Antibiotic, antibody, antigen, or vaccine. We're talking about a chemical substance that stops or kills The growth of disease causing microbe. What is the answer to this? Exactly, it is antibiotic. So antibiotic and antibody ka difference can be done very simply, right? Antibiotic, antibody. So antibiotic, where is it produced? It is produced in the microorganism. Where is antibody produced? Inside our body. This is a chemical substance. We see that this is a protein. Yes, this may or may not be specific, but antibodies are specific. Yes, let's move on to question number 5. Which of the following microorganism plays an important role in maintaining the balance of nitrogen in the atmosphere? So I will discuss nitrogen cycle here. Viruses, bacteria, algae, or protozoa? Very easy. Quickly, students, quickly give me the answer. Very good, all of you, very good. The correct answer is option B, which is bacteria. Now, when you talk about nitrogen, right, nitrogen exists as N2 gas, right? And we know that this right here is not present in the usable form. Yes, so we know that it needs to get converted into a usable form. And this process is what we call as nitrogen fixation. And we know that this process of conversion of atmospheric nitrogen into usable form is done by rhizobium, very good, which is present in the root nodules of leguminous plants. Very good, MLB Dreamer and Saroj and many others. Now, along with this, we also see that there is other organisms like nitrosomonas, we have pseudomonas which are there, right? So, other different kinds of bacteria are also available. Moving on to question number 6. Yes? Paheli dug 2 pits A and B in her garden. In pit A, she put a polythene bag packed with some agricultural waste, while pit B, she directly dumped it without packing it in a polythene bag. She covered both the plants with soil, right? So now, of course, we see that what did she observe after 1 month? Now, waste in pit A degraded faster than pit B. Waste in pit B degraded faster than pit A. Waste in both pits degraded almost equally. Waste in both pits did not degrade at all. Very good, students. Very, very good. I'm so proud of all of you. See, I know sometimes I may not take your name, or sometimes there are comments that I read, but it moves so fast that I'm not able to read the names that are there, right? But trust me, I read all of your comments, and I'm very proud of all of you that you have progressed this much, right? So now, of course, in the soil that is there, we know that there are various microorganisms which are present that facilitate the process of decomposition, and waste in pit B degraded much faster, right? I just told you that I'm not ignoring you. Just that the chat is moving so fast. I try so hard to pick your names from it, but I'm not able to do that. I'm also human. No, I don't have eyes like robot to find names. Yes? Thank you, Anj. Thank you to many others, because you were telling me that you've been following my classes pretty regularly, and I see that it's been helping you improve, so I'm very proud of you, right? Ma'am, may I know the reason? Of course you can, road to engineering. See, in pit A, what happened? She put all of it and she put the agricultural waste, right? Now in pit B, she directly dumped the soil. So when she directly dumped the soil, what happened was that the microorganisms in the soil started to decompose the agricultural waste, right? So that is why we see that the waste in pit A degraded much faster. Now Athar was asking me the organism that causes citrus canker. I think it is Xanthomonas. Xanthomonas, yeah. Xanthomonas citri. I was just making sure I get the second part of it. Xanthomonas citri causes your citrus canker, and the rust of wheat, right? That is caused by a fungi called as Puccinia. Yes. Jairam, you can send me the answer, right? You can send me the question once again. Nancy, hello. Where are these questions taken from? Different sources, right? Different sources. Moving on, everybody. Plasmodium comes in protozoan. Let's go on to the next one. Azotobacter is an example of curd producing bacteria, nitrogen producing fixing bacteria, bacteria used for making alcohol, or immunizing bacteria. Use logic. If you don't know what azotobacter is, use logic. Saurabh sir is the best. Arey, my Hindi is... It's not bad, ठीक ठाक है, but explain करने में थोड़ी दिक्कत होती है. इसीलिए जब मैं explain करती हूँ, मैं English में explain करती हूँ. ऐसे बातचीत करने में हिंदी ठीक ठाक है. But explanation, my Hindi is very bad. I mean, I'm more comfortable, I would say, right? Yes, I will do it. हाँ. नाम ले लो, हाँ हितेन. Thank you, Anushka. B, C. Oh, there is so many... Try कर लो, हाँ try करती हूँ, practice करती हूँ, थोड़ा अभी like see how I'm speaking, right? Yes, LM classes. Thank you, Keshava, thank you. Answer is nitrogen fixing bacteria. Curd producing bacteria, you know it is lactobacillus. We are clear, right? Now, bacteria used for making alcohol, we know that per se it is not there, right? But we know that when it comes to production of alcohol and commercial alcohol, it is yeast, yes. Then you have immunizing bacteria, which is your disease-causing bacteria. So I know there would be a confusion here, which is why for all of you out there, nitrogen-fixing bacteria is or azotobacter is an example of nitrogen-fixing bacteria, right? Now let's move on to last 3 questions, yes? Ma'am, there is only one nitrogen-fixing bacteria and that is Rhizobium. No! Rhizobium is a symbiotic, or Rhizobium is a nitrogen-fixing bacteria which is having symbiotic association with your leguminous plants, right? Now, there are many free-living bacteria also. See, there are land and fields wherein, or there are agricultural field where if I don't have rhizobium, right, or if I don't grow my leguminous plants, even then there is some amount of nitrogen, no? How exactly did this nitrogen come into the picture? Because of the soil bacteria, where some soil bacteria have the ability to do this, right? Purpose of blue-green algae. Blue-green algae is a primitive organism, very ancient organism. I mean, not very ancient, right? But it's a primitive organism and it is one of the autotrophic bacteria that is there. Okay. Benefits. Important question will come in exam for sure. What are the benefits that are there? We know that the benefits that are there, we know that bacteria unicellular, benefits of bacteria, we know that this right here, of course, many good bacteria are present in the intestine, helps in production of antibiotics, important vitamins in our body are, synthesized with, or they are produced with the help of some bacteria. Then, of course, we know that they act as decomposers, right? So you can write 3 to 4 examples, it should be easy. Yes, 153K has happened. Anabena is another example. Anabena is an example of soil bacteria that we find. Thank you. Awesome, you got 5 marks. Actually, there's a very beautiful video which I've done in nitrogen cycle, right? So there's this beautiful video that I've done on nitrogen cycle wherein I've explained nitrogen cycle in great detail. So I'll put the link of that video also. You can watch it. It's like 10 minutes, but aram se I have explained it, right? Ma'am, blue-green algae, yeah, it's also called as cyanobacteria. Yes, diamond lords, you can ask me. Next up, explain nitrogen cycle with a schematic diagram. 5 mark question, short question that is going to be there. See, nitrogen cycle, of course, see, when you're starting nitrogen cycle explanation, start with why nitrogen is important. Now we know that nitrogen is present, almost 78% of nitrogen is present in the atmosphere and it exists in the gaseous form. And nitrogen is necessary by living organisms. You and I need nitrogen because proteins that we need are made up of nitrogen, right? So if you want to synthesize proteins, we need nitrogen. Yes, in the case of plants, if they need to make chlorophyll or if they need to make any important substances, they also require nitrogen, right? Which is why we see that atmospheric nitrogen is necessary for us. But atmospheric nitrogen is not usable. So there needs to be conversion of atmospheric nitrogen into usable forms. And that is where the nitrogen fixation process comes in. So atmospheric nitrogen can be fixed by naturally, or I would say physically, with the help of lightning, where in high pressure conditions we see that atmospheric nitrogen gets converted into compounds of nitrogen, right? So like NO3, nitrates and nitrites that are there, it gets converted into that. or it can happen biologically, right? So it can happen biologically where organisms are there that will fix it into nitrogenous compounds, and then it will go to the soil. Now this soil that is there will be, or this compounds of nitrogen that are there will be taken up by the plants, and we eat these plants directly or indirectly. Now, one other way in which this can be made to the soil is when nitrogen is waste through excretion, like through urine or maybe through excretion of waste materials, it can come, or else it can also be through death, right? Do we get nitrogen cycle for exam? It will come, right? Now, of course, all of us so far we have spoken about taking in the nitrogen. But this 78% of nitrogen remains the same in the earth, whether you and I are using proteins or no, right? So how exactly did this happen? This happens because there is certain nitrogen, there are certain bacteria that are there, right? We see that there are certain bacteria that will convert the compounds of nitrogen back into gaseous nitrogen or N2, right? And it will go back into the air. Who discovered smallpox? That I don't know, but I know who discovered smallpox vaccine. Denitrification. Denitrification is conversion of compounds of nitrogen, that is like your nitrates and all of that, into gaseous N2. Are we clear? Blue-green algae is, cyanobacteria is basically bacteria, right? Robert Koch, very, very important person who played an very crucial role in discovery of various diseases and how to categorize them, right? Dead cells, basically who discovered cell was Robert Hooke, who ended up discovering cork, that is a dead cell, right? Okay. See, now for those of you who have not understood, right, who have not understood a little bit on nitrogen cycle, shall I send you a link wherein you will be able to watch the detailed explanation in the comments of this video, right? So I will do that as well, okay? So that you can watch that video, and in case if you still have doubts, you can let me know in the comments. Diamond Lords, let me know this in the comments. I will definitely do such videos, right? What is NO3? That is nitrates. Nitrates, chemical formula. Okay, awesome. Let me know in the comments. We will move on to the last question. Explain the formation of curd. 3 marks you will get from this. This is very, very important, right? So this right here, when we talk about curd formation, you can start with how milk is converted to curd by the process of fermentation, where of course we know that chemical reaction between lactobacillus, we see that chemical reaction of, let me just write this, right? So in this particular case, you can talk about how milk gets converted to curd. You can write about how first a little bit of curd is added to milk, and inside the curd there is lactobacillus bacteria, right? Now this lactobacillus bacteria will act on lactose, convert it to lactic acid, which will in turn impact the microorganism, which will impact the texture of milk and causes curdling, right? Yeah. Kya hua? What is happening between Mohan and Umar and everybody here? Guys, focus. Yes, Shaurya, I will do that, right? Okay, don't worry, Shantanu. We are here and we've got you covered. And Kushank, yes, I do. I think you're the one who thinks I'm fake during the premiere, but I'm actually real. It's me. Okay, students, with this, I come to the end of today's class. Today's class became very long, 1 hour class, right? So naturally, over a period of time, lactobacillus is there, Prahati, that is how is present in curd, right? So students, with this, we come to the end. I hope all of you found this class helpful. I know today's class went on pretty long, right? But many of you here stay till the very end. That means that all of you enjoyed today's class, right? So now, of course, for many of you out there, we are going to have more practice classes. See, this was just the benefit part. Now we also have the harmful part. We'll be pushing some recorded videos. Then we will do practice and doubt clearing. So it's very, very important that all of you need to come prepared. You need to watch concept bite videos. You should not be like, "Arey, I'll go watch and I'll ask Aishwarya ma'am in live class." So sometimes when we come with the summary, we come with the intent that we have already taught you this, right? So in case if you're coming to the channel for the first time, I know many students would be watching this video, which is why I teach in the beginning no matter what. But nonetheless, please make sure you come prepared for such classes, right? Okay. So if you enjoyed today's class, You should let me know in the comments. Please make sure that you all do not forget to like this video. How else will you know that my students are loving my classes, right? So please make sure to hit the like button on this video. Do not forget to share this video with all of your friends. Tell them that you know what, Ashwara ma'am does this amazing classes. You have to watch it. And of course, let me know in the comments if you have some suggestions for me. If you have some doubts that you want me to answer, I will definitely answer. Ansh will tell me, ma'am, you know what, you have not answered my doubt yet, but I will definitely answer it back. And subscribe to the channel. So thank you so much, students, for being a part of today's class. I will see you soon, but up until then, take care, lots of love, and bye-bye. Good night.